Today, most people in the world are aware that infectious disease research is important, but many don't know that the largest cause of death by infectious disease is mycobacterium tuberculosis, the bacteria that causes TB. MTB has been around for centuries and is still driving the tuberculosis pandemic. Part of the reason MTB has been so successful is down to its evolution. Not only has it evolved to live inside its human hosts for decades undetected, but there are also 7 distinct lineages, or groups of genetically similar strains, worldwide. These lineages are geographically localized, and their evolution has been linked to variations in phenotypes such as virulence and antibiotic resistance. Previous research has found that significant differences in the genome, specifically single nucleotide polymorphisms, or SNPs, can contribute to the increases in disease severity we see in certain lineages. But how does the evolution of these SNPs drive changes in phenotype and therefore disease? In my lab, I have used strains from 3 major MTB lineages to answer this question using an integrated genomic and metabolomic approach. First, I identified the SNPs in each strain, selected those which resulted in an amino acid change in the protein sequence, and used them to cluster the strains. The strains grouped perfectly by their lineage classification. Next, I carried out metabolomic analysis by looking at the final metabolites produced by a cell and clustered the strains by the most significant metabolites. This analysis also grouped the strains perfectly into their lineages, suggesting that the SNP data may be driving the metabolite results. Finally, I grouped all the SNPs with identical expression profiles into blocks and correlated these with the metabolites to integrate the datasets. We found several SNP blocks which significantly correlated with specific metabolites, identifying pathways which are potentially affected by these SNPs. Overall, this approach using established tools has identified links in the genomic and metabolomic changes that have evolved in this pathogen. The novel relationships identified here could help in drug and vaccine development to eradicate TB.